The idea that young blood might reverse aging sounds like a myth ripped straight from a gothic horror novel or Silicon Valley startup pitch, but it's also a real scientific question that has captivated researchers for decades. At the heart of this curiosity lies heterochronic parabiosis, an eerie yet enlightening experiment in which the circulatory systems of two animals, one young, one old, are surgically joined so they share blood. As Frankensteinian as it sounds, this technique has revealed surprising truths about how blood-borne factors regulate aging at the cellular and tissue level. In fact, it might be one of the most convincing biological hints that aging is not just wear and tear, but an actively regulated and possibly reversible process. The foundational experiments date back to the 1950s, but the field exploded into mainstream biomedical interest in the early 2000s, when researchers began reporting that old mice surgically conjoined with young mice showed signs of systemic rejuvenation. In these heterochronic pairs, aged mice experienced improved muscle regeneration, enhanced liver function, faster wound healing, and even better cognitive performance. Meanwhile, the young mice? They started aging faster, which made the conclusion inescapable. Something in the blood was flipping the switches of aging, and it worked both ways. So, what's in this youthful elixir? One candidate is GDF11, growth differentiation factor 11, a protein that declines with age but was found in higher levels in young blood. Early studies suggested that restoring GDF11 to old mice improved cardiac function, increased neurogenesis, and even reversed age-related muscle atrophy. It was hailed as a potential fountain of youth molecule, until follow-up studies muddied the waters, showing conflicting results and suggesting the original assays may have confused GDF11 with its near-identical cousin, myostatin, which actually inhibits muscle growth. Still, GDF11 opened the door to a wider investigation of circulating factors, proteins, hormones, exosomes, and even microRNAs that appear to shape the aging process from the inside out. Other promising molecules include oxytocin, which enhances muscle regeneration, TMP2, a protein in human umbilical cord plasma that improves learning and memory in aged mice, and klotho, a hormone-like protein associated with longevity and cognitive resilience. But it's not just about what the young blood gives, it's also about what the old blood removes. Aged blood contains higher levels of pro-inflammatory cytokines, oxidized lipids, and senescence-associated secretory phenotype, SASP, molecules, which poison the regenerative capacity of tissues and promote chronic inflammation. When these are diluted, either by infusion of young plasma or by filtering the old blood, a technique being tested as plasmapheresis, cells and organs seem to respond by acting while younger. This has inspired a modern twist, young plasma transfusion, an approach being tested in both mice and humans. In early trials, Alzheimer's patients receiving plasma from young donors showed modest improvements in functional scores and mood. Companies have popped up offering young plasma therapies for rejuvenation, though with eyebrow-raising ethical concerns, limited oversight, and a sharp divide between hype and reality. The FDA has repeatedly issued warnings against unapproved vampire therapies for anti-aging purposes. Still, the science remains tantalizing. Parabiosis and its offshoots suggest that aging is at least partially programmable, and that the program runs in the bloodstream. But questions remain. Is it one molecule or many? The evidence so far points to a complex cocktail of factors, rather than a single rejuvenating youth hormone. Does it work long-term? In mice, some rejuvenating effects are temporary or come with trade-offs. We don't yet know the full cost of long-term plasma manipulation. What are the risks? Infusing young plasma into old individuals could have immunological consequences or trigger unpredictable responses. Can we isolate the good stuff? Instead of whole blood, researchers are now trying to identify and deliver specific components, such as exosomes, peptides, or metabolically active proteins, that provide the benefits without the baggage. One particularly exciting area is epigenetic age reversal. Some studies have shown that after young plasma infusion or blood exchange, epigenetic clocks, which estimate biological age based on DNA methylation patterns, in old mice actually turn back. If verified in humans, this would be nothing short of revolutionary. Evidence that the biological signs of aging can not just be slowed, but reversed, at least partially, through manipulating blood chemistry. Of course, The idea that young blood might be a literal fountain of youth has uncomfortable implications, both scientifically and ethically. It raises questions about exploitation, access, and what kind of society would normalize transfusions for beauty or performance. But beneath the headlines and hype, the underlying science is very real and incredibly promising. So, is blood the fountain of youth? Not quite, but it might be the faucet. And as researchers turn the handle, filtering, analyzing, and refining what flows through our veins, we may eventually learn how to distill from that crimson river a set of tools to slow, halt, or even reverse the molecular clock. Until then, heterochronic parabiosis remains a strange but illuminating peek into one of aging biology's most provocative mysteries.